Assalamualaikum and salam sejahtera. Welcome back to another online lecture session. Today we are going to proceed lecture number 4, which is chapter 3, cell division. In this chapter, we have 3 subtopics: a cell cycle, mitosis, and meiosis. Definition: cell cycle is the sequence of events that take place between one cell division to the next cell division. It consists of two main phases. First is interphase, which is the longest phase of the cell cycle, and followed by mitotic phase, the shortest phase of cell cycle. Interphase of cell cycle has three main phases. First is G1 phase or first cap. In this phase, cell will grow and develop, where the cell will increase the synthesis of organelles such as mitochondria and ribosome. Next is cell will increase the volume of cytoplasm, synthesize enzyme and protein for DNA replication that will be used in the next phase. And the last one is chromosome in the chromatin form, which is loosely packed. After the G1 phase, cell will enter the second phase of interphase, which is S phase or synthesis phase. In this phase, DNA synthesis will occur. As we can see here, this is one copy of chromosome, and then after replication occur, DNA become two similar copy or known as sister chromatids. For the third phase of interphase, which is G2 phase or second gap, cell will further synthesis of organelles and duplicates the centrosome. As we can see here, from one centrosome to become two centrosome. The last one is mitochondria are highly replicates to increase energy storage of cell. In animal cell, it occurs by a process non-cleavage, occurs through formation of a structure cleavage furrow. On the cytoplasmic side of the furrow is a contractile ring that consists of 18 microfilaments associated with the protein myosin. So the 18 microfilament interact with the myosin, causing the ring to contract. And finally, the cleavage furrow deepens until the parent cell is pinched into two completely separated cells. Compared to the animal cell, cytokinesis in plant cell occurs through the formation of cell plate. In plant cell, vesicles containing cell wall material produced by Golgi body will align at the metaphase plate. Vesicle enlarge and fuse to form a new cell plate. Cell plate grows and unite with existing membranes, forming a two new plant cell. First terminology is chromatid. From this diagram, we have one chromatid and another one chromatid. So two chromatids that attach to the same centromere we call sister chromatids. So centromere is the region in which two sister chromatids attach to each other. The next terminology is homologous chromosome. Homologous chromosome is a pair of chromosomes that are identical in three aspects. First is size, second is genetic information. From this diagram, we can see these two chromosomes has four same genes represented by allele G, R, S, and T. The third one is same in terms of position of centromere. So we can see here, the centromere of first chromosome is the same level to the next chromosome. 4 is synesis, the process of pairing homologous chromosome during prophase 1 of meiosis. This is one homologous chromosome. So the process of pairing we call synesis. Okay, next is bivalent, a pair of homologous chromosomes which undergo synesis. So we can label this structure as bivalent. Next is tetrad, a group of 4 chromatids of a pair homologous chromosome which undergoes synesis. So this structure also we can label as tetrad if we refer to the number of chromatids 1, 2, 3, 4. Terminology number 7 is crossing over, a process of exchange genetic material between non-sister chromatids of homologous chromosome during prophase 1 of meiosis. From this diagram, we can see this is one homologous chromosome. This is non-sister chromatid between blue and red, and then this is process crossing over. The significance or importance of crossing over, okay, first, result in genetic recombination. Second is increased genetic variation in especially gametes. Okay, the last terminology is chiasma, the regions of crossing over that occur during prophase 1 of meiosis between non-sister chromatids. So here is a process crossing over, and the place or region where the crossing over occur we call chiasma. Let's take a look at the overview of stages in meiosis. In meiosis, there are meiosis 1 and meiosis 2. So for meiosis 1, we have 4 main phases, which is prophase 1, metaphase 1, anaphase 1, and telophase 1. For prophase 1, this is the longest phase of meiosis. It divided into 5 subphases, which is leptotin, zygotin, Pakitian, diplotin, and dikinesis. Whereas for meiosis 2, you also have 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2.